Hello, this is Rich Eisenberg. I'm the editor-in-chief of Inorganic Chemistry, and as part of our special 50th anniversary year, we have interviews with a number of leaders in the field. And today our special guest is Professor Edward Solomon from Stanford University. Ed is a distinguished scientist in the area of physical bio-inorganic chemistry, and also has the distinction of being the longest-serving associate editor for the journal Inorganic Chemistry. We're going to get to that in a little while, and now let me say welcome, Ed.
Thank you, Rich.
Ed, we've known each other for quite a while, and I know well what you do, but for our audience, can you tell us a little bit about how your early education and training influenced you in terms of the research that you currently do?
In terms of how I started in chemistry, I grew up in North Miami Beach, and I've probably been a scientist my whole life, basically. My parents went to New York on vacation, took me with them, and we visited a planetarium and museums, and the science just got to me. When I went home, I got a telescope and a microscope, and and eventually Gilbert chemistry set, which expanded into our garage and took over the garage. And I somehow got into chromatography in junior high school, and I think reading a Scientific American article about Michael Spaght, and entered and won science fairs in chromatography. And then in high school, I got in a program where a few students would get into working in laboratories. I was in Dade County, and they started this program where I worked in a laboratory at the University of Miami and at a medical research center, and started working with indoles and schizophrenic patients, and eventually was the first person in Florida to win the Science Talent Search, a national thing, and just kept going in science. So it was, so I just guess I've always been a scientist.
Great.
And a chemist.
Now, tell us how what you did when you were getting your PhD, and what you did before you assumed your independent career.
I guess in undergraduate, the things I really liked were 10DQ and the Jahn-Teller effect, and spectroscopy and quantum mechanics. And so I recognized, after looking into possibilities for graduate school, Don McClure, who had just moved from Chicago to Princeton, was the person I wanted to work for. So, I went to Princeton, did my graduate work with Don. And during the time I was there, Don and Tom Spiro organized a really interesting symposium that had all the leaders of physical and organic, going from theoretical physicists to very synthetic inorganic person. And I looked at the list and said that's what I wanted to do in postdoc, which was working with Harry Gray. And so that's how I decided to go in that direction, because they were really interesting molecules, and they seemed like they'd be a lot of fun to study.
With regard to getting into inorganic chemistry, I remember that colors were always a fascinating thing for me, but you said 10DQ and Jahn-Teller effect, but I didn't know about Jahn-Teller effect. Was it the spectroscopy of the compounds, the colors of metal ions in solution that drew you to ligand field theory?
Way back through junior high school, I had rock collections and mineral collections, and so always have been interested in that. But just the simple concepts as an undergraduate, that I guess eventually led to an interesting group theory, but they weren't quite taught that way then. Just seemed like electrons in d orbitals were what I wanted to work on, and with Don it was pretty chemical physics related and solid state physics, but I thought I wanted to really evolve more to interesting molecules, which is how I went from more of the solid state physics to, or chemical physics to the inorganic direction via Carl Ballhausen in Copenhagen, because I wanted to make the transition from Don, who was really a spectroscopist who sort of worked right at the edge of spectroscopy and theory, to Carl Ballhausen, who was a theoretician dealing with experiment with spectroscopic results. Plus, he was more molecular, and also has a chance to live in Denmark, which really was a very interesting time. When I worked with Carl Ballhausen, one of the things I focused on, the main thing I focused on, was the red band of hexaquanickel and understanding why this absorption band had a double peak. And this was a big controversy between Carl Ballhausen and Kliksbo-Jorgensen that led to their going anti-bonding with each other. Anyway, this was what I worked on in Denmark. And so when I went to Harry's lab, I went there with the idea. In fact, I worked both on trying to predict what the structure, which we pretty successfully did, of the blue copper active site and proteins like plasticinin, but also I worked on quadruple bond spectroscopy. And the thing that really let me make the transition, I think, conceptually into metals and biology was Harry had a spectrum of nickel substituted carboxypeptidase, an absorption spectrum, and you look at it, and it basically, you can map concepts from the red band of hexaquanickel, or the spectrum of it, and start understanding that metal sites and proteins were basically unique inorganic complexes, and you could start thinking of it as an inorganic complex, but with this ultimate ligand that leads to all this interesting chemistry. And that's how I really started in the bioinorganic. And when I went to my career at MIT, I had, as a graduate student, been very interested in these copper magnetostructural correlations of people like Bill Hatfield in the 60s. And it really amazed me that there were copper sites, binuclear copper sites in proteins that had completely different physical properties of anything people had seen before, and they did chemistry that nobody can accomplish in inorganic chemistry. And so that's what I decided to work on, as a chance to really go back to binuclear copper chemistry, but in protein sites where everything is new. And that was my evolution. And originally I also started projects in understanding inorganic photochemistry, Adamson's rules, in terms of excited state potential energy surfaces, and also looking at organometallic ligands on metal oxide surfaces, Werner surfaces, to understand properties related to heterogeneous catalysis. But as my research evolved, it just all went into different problems in bioinorganic systems, because they are so fascinating.
Now, these threads continue to run through your research even today. Describe a little bit about how you view your research, the themes that you set for it.
Well, the themes are electrons in d orbitals, lots of types of spectroscopy, and developing new types of spectroscopy, and understanding how electronic structure leads to reactivity.
What would you describe as, let's say, your most satisfying or proudest accomplishments in this area? And there have been a lot.
You had me think about this, and I think there are 5 that I would go through, which are not necessarily in order, but my earliest research really dealt with, on my independent research at MIT, dealt with the fact that we showed that the binuclear copper site in tyrosinase was basically the same as the hemocyanin, that oxygen was activated to the same level, but tyrosinase is an enzyme, and we showed that's because the substrate can access and bind to the metal site, and that led us to generating a mechanism for how the enzyme works, which is still the one that is always invoked in the field. We showed a trinuclear copper site exists in biology. When we did this, it was so unaccepted that I almost lost my funding. Actually, we showed this in 1985, and my grant went up to a panel and said it was impossible that the type 1 and type 2 are here and a binuclear site is over here. And eventually crystal structure in 1989 showed that the spectroscopy was clearly correct. But we also, over the years, trapped the intermediates in the reaction mechanism and could see how nature reduces oxygen to water and with very little overpotential. And in blue copper, we've shown how the protein controls the geometric and electronic structure of the site and can tune reactivity. This is this concept in bioinorganic called the enstatic or rack state. In non-heme iron enzymes, these have been generally viewed as spectroscopically inaccessible. They don't have the porphyrin that gives it a lot of color, and they are even electron ferrous, high-spin ferrous sites, and so we developed this variable temperature, variable field, magnetic circular dichroism, VTVH-MCD approach to study these. We developed the analysis of the type of data and it really opened up our ability to study these enzymes, and the studies define a general mechanistic strategy used by all of the different classes of non-heme iron to turn on oxygen reactivity and generate intermediates only in the presence of all the co-substrates. And the last of these that I want to bring up is we developed spectroscopic methods, metal L-edge and ligand K-edge spectroscopy that let you experimentally look at the covalency of ligand-metal bonds. And we've applied these to a variety of problems in iron, sulfur, and copper, sulfur electron transfer sites, and now have been looking and starting to understand diphylline molybdenum bonding related to oxotransfer.
Now, this is a little different. Sounds similar in question, but it's a little different. Which is, were there any what I'll call eureka moments where you did something or a result was obtained and you knew that either you had opened a whole new area, cracked a problem, achieved a deeper understanding, something of that order?
Well, yes. Yeah. I'm sorry. My first eureka moment, I guess, which was as a graduate student, which is the only reason I went on to an academic career, was based on the research I was doing with Don McClure, where I was studying the lowest excited state of high-spin D5 manganese and rubidium manganese fluoride, and I would grow single crystal in a bridgement furnace and orient it and cut it and polish it and then put it in a piston and squeeze it in the middle of liquid helium and shine light through it to take spectra. And I was looking at the lowest excited state, and I could see 3 substates all split by 6 wave numbers in the absorption spectrum of this complex. But when you took theory and all the existing theory and put it together in terms of writing a Hamiltonian, it would calculate a splitting of 100 and some odd wave numbers. And the more terms you put into it, the worse the agreement between experiment and theory got. And so at some point in my graduate career, I decided I just had to see if I could figure this out. And so I first surrounded myself with the data and made sure all the data were the way they were, but then I got into the theory and sort of wandered around Princeton campus for a few days just thinking about terms in a Hamiltonian, and is true, and realized that the way people have been doing things at that point missed a certain point about how these terms can be combined, and they really weren't being done right. And figured out what I thought should be done, went to the computer, which in those days was an IBM 360 underground, and I went and put the computer cards in, and as soon as the calculation came out, it was 6 wave numbers, and agreed would experiment. And also, when I realized this model, I could, I could use it in different ways to correlate with different types of data I had obtained on these 3 origins over the years, and it explained everything very quantitative. And at that point, I knew I could solve problems, and I, felt confident to go on and, actually end up in an academic career.
Yeah. Now, your papers have always been really, ideal examples of scholarship and deep thinking. In terms of suggestions or comments to young scientists today, what would you tell them in terms of communicating their science?
Well, I guess I think the important thing is not to fragment and try to publish it in small pieces. I believe you should really stick with the science and figure it out and do the tests to make sure that your model is right, and that you should be your own biggest critic about the science. Your goal isn't to get it through a reviewer. It's to put all this effort in with yourself and your students. You want to make sure what you've done is right. I know there's a lot of pressure on junior faculty to publish quickly, and it's real, but in dealing with this, you don't want to, you want to stay by your standards on what you're looking for from your science.
One of the things that is a hallmark of your research career over the years has been a lot of collaborations. How did you develop, and how do you develop these kinds of interactions and collaborations?
I think that's one of the really fun part about doing science. You just, sure, you can spend months isolating an enzyme and going through this process. And usually what we do is choose a few systems we're going to study and develop a method in, and we will isolate them ourselves and and develop an approach. But once we have it, it's so much more exciting to collaborate with people who are experts on their topic, and science can move quickly where we bring in our expertise. And the person or people we collaborate with have their expertise, and just, you can do so much more. And I think, with the way travel is and the way, we can move samples around, the data around, it's just, science now should be collaborative and internationally collaborative. It's only a really positive thing. There's these unique molecules that are inorganic and biological, and we want to study. I mean, I still study ones that are in the context of the things we think about. So we don't pick up, just any study. But if it's in the scope of things we're already really focusing on, we can only much more leverage our science by collaborating.
Ed, let me ask you to project ahead, and in terms of the direction of chemical research, and at the same time the enduring qualities that first-rate science always needs to have.
Yeah, I've thought about this, and I think that as we all know, instrumentation is getting more and more sophisticated. Computer power is following Moore's law, and the level of computational resources, quantum codes, are getting more rigorous and more accurate. And so I think it's our jobs as scientists to really make maximum use of these incredible resources as they are available, and to use them to go to places we haven't been able to go before. And so, from just my perspective, for example, we're going to have an X-ray free electron laser at Stanford next year, and I fully intend to use it and to do experiments that I think will be relevant to apoptosis and may even relate to photodynamic therapy. And so I think you've just got to make sure your science is maximizing what you can do with the new resources as they become available.
Today there seems to be a great push to applications in the research that gets funded and the research that people are doing. What advice would you have for young investigators today in terms of how to frame the problem that they work on?
Yeah, well, when I was a graduate student, as I said, I spent a lot of effort working on the first 6 wave numbers of the lowest excited state of rubidium manganese fluoride, and put a lot of effort in, and I could solve it, and I wrote a very satisfying 30-page phys-rev paper on this, and I'm sure there were a few labs in the world who read it carefully, but to me it's if you're going to put that effort in, you really want to communicate your science with a very wide range of different types of scientists. If you're going to do that effort, do science that's going to overlap with a lot of different areas. And so I think it's fun to do science that maybe is fundamental but does have applications. Based on some some papers we published in one area, it amuses me the range of scientists that oncologists and microbiologists who want to know what a non-Cromers ion is. I mean, that's fun to have people in those areas reading your science. So I think as a physical and organic chemist, you can choose a lot of different problems. So why not focus on problems where what you learn is of importance to a lot of different people and does have impact in life sciences and catalysis and materials.
Well said. How many students have you mentored over the years, both for PhDs and as postdoctoral researchers?
I've had a little over 100 students, 2/3 graduate students, PhDs, and 1/3 postdocs, and about 45 in academics around the world now.
That's terrific. But it also takes a lot of work to feed all the mouths when you have them in your lab and with proposal writing.
The proposal part is the stress of doing science. When I started my career, I thought the hard part would be to start build a lab and build a research program and accomplish science and get it funded in the top journals. And instead, I learned all the stress is really getting grant writing and getting funds. And we're really entrepreneurs who have to not only figure out science and sell it to our peers who are on the panels and the study sections and the program directors at the different funding agencies. And you not only have to convince them it's worth doing, but you have to convince people it's in the top 10% or so of this very good science that they're evaluating. So I think to go into academics, in addition to wanting to, or at least research-oriented academics, in addition to wanting to mentor and teach, you really have to have areas of science you want to work in and that you're driven to solve problems to make it worth all the stress of trying to get money through funding your research. Okay.
I'm going to turn to a different subject now, maybe a little less stressful than grant writing, but I would say it's stressful, and that's editing and inorganic chemistry. As I said at the beginning, you are the longest tenured associate editor of inorganic chemistry.
Is this a tenured position?
Yes. But my question is, tell us how you got into this at the beginning, because you started it very young, and how did the journal work at that time, and your perceptions of how we work today?
Yeah, I started in 1985, right after I got back from Bali from my honeymoon, actually, and Fred Hawthorne had asked me to step in to the position, and at that point, Fred was the editor-in-chief, and he handled the main group papers, and Herb Case handled the organometallics. Steve Lippard, bioinorganic, Jack Williams, structural papers, and I took over for Galen Stuckey on the bioinorganic side. And that was pretty much how it was over the years till you took over in 2001. And it really changed at that point. We went from the bright yellow cover, which was useful to meet seminar speakers at airports, but to a more pleasant but not perfectly satisfying green color with a picture on the cover emphasizing one of the papers in it, which has become, I think, very attractive to a lot of researchers wanting to get their science on the cover. And we got rid of notes which were low impact and went to forums which were very high impact. And I guess also the board of the associate editors expanded to 9, which I think just reflects how interdisciplinary the field is, and also the huge volume of papers we get.
I was going to ask you, what could you say about the papers that you used to get when you started versus the papers that you get now? Things have changed a lot, and as you know, structural work now can be done in an afternoon, whereas it used to take months. Calculations are done in several hours, even by people who are not true theory experts. So the scope of problems has expanded while the techniques have gotten much easier to use.
Yeah, well, yeah, I've viewed there being interesting evolution of what I felt my goal was in the physical inorganic and bioinorganic evolution of that part of the journal. Originally, it was to attract the strong spectroscopy papers back to inorganic chemistry. They had really gotten diffuse and more into the chemical physics and the molecular spectroscopy literature, and so it was really get the spectroscopy back into inorganic chemistry. Then there was a period where there started to be a number of bioinorganic journals, and we had to make sure IC stayed the premier journal for publishing the detailed model type studies, but also to attract the physical and chemical studies on the metalloenzymes into IC, which I think we've done very successfully. We had about 10 years ago the burst of DFT starting effects, which density functional theory, which meant we had to establish, and I think we were the first general journal to do this, establish criteria for what type of theory papers belong in inorganic chemistry, which really were chemically relevant with insight, and if they did get published in IC, we also wanted certain supporting material so people could actually evaluate the calculations and reproduce them if they had to. And I think, as I said, we probably were the first journal to establish these. And to me now, I think that one of the things I view as a problem is how like everything's DFT now in physical and organic chemistry. The it used to be 10-90 for theory to spectroscopy and experiment, but now it's like 90-10. And it worries me because spectroscopy is the way it is. That's experimental electronic structure, and people should realize that you can get a lot of different areas with different results with different type of calculations, and it's really important to test these with experiment. And once calculations are supported by spectroscopy, they can go further into, let you extend further in terms of understanding more about the bonding and even going into reaction coordinates and looking at transition states, which you're not going to study experimentally, but spectroscopy is reality, and it's important to have calculations based on this.
let me ask you, this is, as we embark in this 50th anniversary year, the particular challenges that inorganic chemistry faces today, and what it is an increasingly diverse set of papers directed towards perhaps particular target audiences that are outside of our traditional boundaries. How do you view A, the challenge to inorganic chemistry, and B, the special opportunity that inorganic chemistry offers for researchers in those areas?
Well, I guess I would answer you by saying the way I choose to put my own science into inorganic chemistry is to do put in fundamental, in-depth, rigorous studies that might be longer and more technical than for JACs, but really core science that does overlap with a lot of different areas. Just as an example, recently I published a paper in IC on spin-frustrated trimers and getting into experiment and analysis of things like antisymmetric and anisotropic exchange, and I thought it was really nice studies on these systems, but it was also very relevant to me because the same phenomena we were looking at, it was important to the native intermediate and the multi-copper oxidases, but it's also important to spin-canted magnetic materials. And so I think the type of science overlaps a lot of the range of interdisciplinary areas. And I think, so that's why to go to IC. And I think dealing with the range of interdisciplinary areas by having expanded the associate editor group, you've really expanded the scope of what we have the competence to handle well in terms of the journal. So, I think we're doing, really well at staying state of the art.
Yeah, you had mentioned before about forums, and I should point out that you were one of the people that came up with that particular idea, and I think it's been very successful in getting interest from both within the inorganic chemistry community and outside the inorganic chemistry community on certain theme topics that, and I know you've done and edited a couple of those.
No, I think the forums are great because we're looking at areas in a different way than a lot of past attempts, and we're also pulling in people from areas that wouldn't normally publish in IC. I did one forum that I had all the authors actually were people who had not published in IC before. They were young physical methods and metalloenzyme systems, and your forum on oxygen evolution brought in a quite diverse group of people.
As we get ready to go electronic fully, and now you won't have anything to hold up in an airport to greet visitors, what do you think?
Well, I think, as we were discussing recently, I think going electronic is a lot better than the recent very small copies, which are very hard to read. And so I think if we're going to do it, we should just get there. And our students are there already. I mean, they're not going to be doing hard copies, and they take a lot of space. So I think it also keeps the science flowing very quickly. As it's, the ASAPs, as soon as they're out, that's when people read the literature.
Right. So old dogs like us are going to have to Twitter a lot.
We'll see. We'll see.
All right.
Do you Twitter?
I haven't yet, but I'm going to tweet. Okay, tweet, right. Okay, with that, let me thank Ed Solomon for this interesting interview. And
thank you, Richard.